Okay, it looks like we're live. I'm your host, Conrad Cording, and I am so excited about three wonderful short talks. We will first hear from Scott Rich, and then from Maria Schneider, and then from Francesca Castaldo. And we will start with Scott Rich's talk. I'm looking forward to it. Go for it.
Awesome. Thank you very much, Conrad. I'm really excited to sort of kick off these short talks here at Neuromatch 4.0. Today, I'm going to talk about work entitled Resilience Through Diversity, Loss of Neuronal Heterogeneity, and Epileptogenic Human Tissue Impairs Network Resilience to Sudden Changes in Synchrony. Titles has never been one of my strong suits, so I apologize for that. And this is up on bioRxiv. So to start off, I want to briefly motivate why our group is so interested in epilepsy research. And it's simply because it is the most common serious neurological disorder in the world. It's really interesting that epilepsy has been characterized to some form or degree going back to the dawn of modern medicine, to the ancient Greeks and Hippocrates. And perhaps because of that, we've had somewhat effective pharmacological interventions for this disorder going back more than a century. Obviously, the primary symptom of epilepsy is recurrent seizures, seizures being this hyperexcitable, hypersynchronous brain state that can yield a wide variety of effects, most commonly these convulsions that can be quite debilitating. And if you look at these pharmacological treatments, a couple of interesting things pop out. One of these is that there seems to be, sorry, I'm just going to throw my laser pointer up here. So one of these is a sort of hard ceiling of about 2/3 of patients to become seizure-free with these treatments. And the other is that there seems to only be, one underlying strategy for all of these drugs, despite, decades of drug development. And the strategy is the hypothesis that seizures arise from excess excitation. Now, this makes a lot of intuitive sense, with seizure being a hyperactive brain state. It makes sense to stop that. We might want to tap on the brain's brake pedal by increasing inhibitory signaling, or ease off the gas pedal by decreasing excitatory signaling. But if this were the be-all and end-all pathway to seizure, we would expect these drugs to be effective for much more than 2/3 of patients with epilepsy. If we want to come up with treatments that are effective for those 1/3 of patients who are medically refractory, who don't respond to these treatments, we need new hypotheses underlying the pathway to the seizure state that we can then target and disrupt with potential medical interventions. Now, one interesting idea that we found in this pursuit is the topic of neural heterogeneity. Heterogeneity is a preserved feature throughout the body, and it actually serves an important physiological role in the brain in facilitating information coding. There's a wide range of sources of this heterogeneity, and what we found digging into the literature is that a lot of the varying etiologies of epilepsy can be recontextualized as losses of heterogeneity. The loss of specific cell types, often this occurs with specific types of interneurons, can be a cause of some types of epilepsy. That can be thought of as a loss of heterogeneity amongst the interneuronal inhibitory population. The misexpression of ion channels, which underlies other types of epilepsy, can have the effect of homogenizing the intrinsic properties of even similarly classified cell types. And then finally, this process of synaptic sprouting, which tends to accompany seizure, particularly seizure that comes about due to traumatic brain injury, homogenizes the input to the two various neural systems. Viewed together, this led us to hypothesize that epileptogenesis can be recontextualized as a progressive loss of biophysical heterogeneity, rendering neural circuits less resilient to the onset of seizure-like dynamics. And what we think is really intriguing about this hypothesis is it's a direct relationship between heterogeneity and a neuropathology, something that hasn't been directly studied as of yet. Because of that, we wanted to take a very interdisciplinary approach to this problem, bridging clinical, experimental, computational, and mathematical neuroscience. This all starts right in the OR. One of my supervisors, Dr. Taufik Valiente, is a clinician scientist. He is performing surgeries on medically refractory epilepsy patients and resecting tissue to reduce their seizure load. That resected tissue goes straight to our wet lab, where arguably the greatest patch clampist in the world, Dr. Homera Moradi-Chamay, is able to do patch clamp recordings on live human cortical neurons, which is extremely, extremely cool. We're lucky enough to be one of few labs in the world that's studying human neurons with this level of micro detail. As a computational modeler, I can then take those micro level details, implement them in networks, and see what the connections are between micro level single cell features and the network level dynamic, which is a seizure. And then, with the help of my other supervisor, Dr. Jeremy Lefebvre, we're then able to reduce this down into a mathematically tenable system and do some mathematical analysis to get at the underlying mechanisms here. And what you'll see towards the end of this talk is what we think is really cool, the sort of closing of this experimental theoretical loop, this cool theoretical result that actually explains some experimental results that were initially somewhat confounding. Now, obviously, for this hypothesis to hold any water, it needs to be true in the biophysical reality. So what we did is we quantified the excitability of neurons via a very simple distance to threshold measurement, just the neuron's threshold potential minus its resting membrane potential. We characterized this both in two non-epileptogenic control populations and also from neurons taken directly from the clinically defined epileptogenic zone, the zone that is responsible for generating seizures. This is really cool. This is one of the first characterizations of the properties of neurons, of human neurons directly from the epileptogenic zone. And lo and behold, what we found is that the variability in this distance to threshold measurement was significantly reduced in the epileptogenic tissue. You can probably appreciate this more easily via the Gaussian fits to this data. And what we're going to do for the rest of this talk is extract these sigma values, these standard deviation values, and implement them in our computational model. So when I talk about low epileptogenic heterogeneities, I'm referring to the low heterogeneity we see amongst our epileptogenic population. A high level of heterogeneity comes from this temporal non-epileptogenic population. So I won't go too much into the guts of our model just for the sake of time, but all that is up in the bioRxiv. I'll mention simply that we use a Poisson neuron model where the primary parameter is a direct analog of this distance to threshold, which allows us to have a one-to-one correspondence between our experimental data and the heterogeneities that we model. We look at the very basic stereotyped EI network and look at, again, high and low levels of heterogeneity, where that is the only parameter that we're changing. When we have this healthy high level of heterogeneity and we linearly increase the drive to the system, we see that the network dynamics, be it the synchrony, the excitatory firing rates, or the inhibitory firing rates, scale more or less linearly with the drive. If you were to think about what this would look like in some sort of LFP analog, you probably wouldn't see all that much difference beyond a little bit of uptick and spiking. Contrast that with what we have in this low heterogeneity case. Here we see this very abrupt, sudden transition from sparse asynchronous firing into a highly synchronous state that corresponds with this increase in both excitatory and inhibitory activity. This is very highly stereotyped. It occurs very reliably at this level of drive. And again, if you were thinking about an LFP, this would essentially be a transition from a sparse asynchronously firing state into some high amplitude oscillation, which obviously has parallels to the hypersynchronous hyperactive seizure state. Now, if you're looking at this critically, you might say, well, this is just best anecdotal evidence from this one particular type of network, that this decreased heterogeneity makes the system less resilient to seizure-like activity. I might disagree with that analysis, but if I'm going to take you at face value, I can then go to the mathematics to convince you that there's actually an underlying theory explaining this phenomenon. Our networks are reasonably simple enough that we can do a mean field reduction to get this into a system we can analyze mathematically. Again, all the nuts and bolts of that are in our bioRxiv. But again, when all we do is change the heterogeneity levels, we see completely different mathematical structures. The high heterogeneity system will always have a single stable fixed point regardless of the external drive. But the low heterogeneity system is much more interesting. At low levels of drive, it'll have multiple fixed points. We then undergo a saddle node bifurcation that leaves a single unstable fixed point. For those who are more mathematically inclined, you may know that saddle node bifurcations are very commonly associated with the onset of seizure-like activity, oscillatory activity, which has clear parallels to seizure-like activity. And what's really cool is that the level of drive at which this bifurcation occurs corresponds almost exactly with the level of drive where we saw the transition in our spiking Poisson network, showing the correspondence between the spiking network and the mean field. So all of this is, we argue, very strong support for this new hypothesis. We've shown that this loss of heterogeneity exists experimentally in human epilepsy. We've used our computational models to show that the loss of heterogeneity has an effect that's associated with seizure onset. And then we've used the mathematics to uncover a fundamental explanation for this phenomenon. Now here's this capstone that I was foreshadowing earlier. And this actually has to do a little bit with something we heard in the morning keynote talk. We actually found that our epileptogenic neurons, on average, tended to be less excitable than their non-epileptogenic counterparts, something that's somewhat surprising because we think about epilepsy as a disease of hyperexcitability and seizure as a hyperexcitable state. But when we look back at the math, we realize that this made some sort of sense. If we average out a more heterogeneous population of activation functions, these fainter blue curves in the background, the population activation function, this bolder blue sigmoid, becomes more flat, more linear. This ends up lifting up the activity values at low levels of drive that best correspond with the FI curve. And obviously the inverse is true when there's low epileptogenic levels of heterogeneity. And lo and behold, I can just plop that experimental FI curve onto this mathematical characterization of the effects of heterogeneity and see this really powerful correspondence. What this allows us to say is that you can actually have a population of neurons that is hypoexcitable, that would be less excitable in the interictal between seizure states. But because that population is less heterogeneous, that will make it more prone to hyperexcitable network phenomena, hyperexcitable oscillatory phenomena that could be either seizure or an associated oscillatory dynamic. So with that, I'd like to thank both everyone on this project and also my third co-PI, Dr. Francis Skinner, of course, our funding sources, and I'm happy to talk about any questions you guys might have.
Cool. Thank you for the great talk. I will be clapping for everyone else that you can't hear. The roaring applause, it's one of the things I'll forever miss. Here's a few questions. So here we have a question by Ji-Hyun Shin, who asked, have you measured heterogeneity in other biophysical
Yes. So
elements?
the answer to that is yes. That is sort of a very ongoing element of our experimental explorations. It's made very difficult by the fact that it's quite hard to get this epileptogenic tissue. That is significantly rare just from a clinical surgical perspective. And in the human setting, we are obviously limited to what we can ethically obtain during surgery. There are other parameters that show interesting patterns with this heterogeneity, in particular with regards to the activity and the expression of the H channel, which is known to play a role in various types of epilepsy. We kept this particular study quite limited and focused because we had, this experimental data that was statistically significant that we could implement. But that is definitely a major subject of ongoing work.
Cool. So as a last question, I want to take the question by Rondi Tuyo, who asked, could you comment whether these results provide insight on why medial temporal lobe tends to be a focus point for seizures in many individuals? It's a great question.
Oh, that is a good question. I'm not sure if I have a good answer for that.
I mean, like, to push in that direction, like, I can give you a reason why primary visual cortex is not the location, because like all neurons are heterogeneous kind of by definition because they sit at different places and so forth.
Yeah. So that's a lot of the work that looks at heterogeneity. It often looks at like metrial cells when it comes and all these things that need to be heterogeneous in order to process information. So it could be just that. It could be that these cells are slightly less heterogeneous on average, even in the physiological state, that makes it more prone to that activity. But yeah, that is a very interesting question that, again, I would love to have more control and more data from all these different regions where we could explore that. I'd have to go become a neurosurgeon to be able to handle that myself. Unfortunately, I don't think that's happening.
It's a wonderful question. So let's
It really
again, and celebrate the great talk.